Okay, guys. Let's do this. Come on. Okay. So make sure make sure all groups are well connected by email and phone and whatever needs to be for this gene expression project, because it is important. And if I see that people aren't working hard, then I might increase the value so that the people who do work hard will get more for it. That's all. Okay. You guys gave me nightmares last night. I just want you to know. I'll get to that in a minute. So exam 1, exam 2, graded by Friday. I don't even know what day it is. Gene expression topics are due this week. And there's no paper investigation, just an exercise. Those of you who've been through it, you know that. Those of you who are coming today and Friday, now you know. So quick note about exams and why I had nightmares last night. I grouped all the exams by which essay question we did. And there was one theme throughout so I got through all of the telomerase ones, which was the majority. There was one theme, which is that people are confused about the function of a primer and a template. People who shouldn't be confused. People who I'm very disappointed are confused. So, and I actually did have dreams about this, which is really bad. But that's probably because I did it while I was going to sleep, as I was. Anyways, so I thought I might start. I wanted to clarify this because you guys cannot graduate as biology majors not understanding what a primer does and what a template is when you're talking about telomerase and DNA polymerase. That is not allowed. I will not let it happen. So I'm going to take a moment here. Because then you're going to go off and use bad language, not bad language, but bad molecular biology language, and people are going to say, where did you go to school? And then they're going to say Towson, and then people are going to go, oh. And that's not good. We want people to go, wow, when you say Towson. Okay. The definition of a primer by dictionary.com. Okay, so you should have no confusion, because if you think about the meanings of the words, you should never, ever confuse them. Now, I must have graded 35 exams where people answered this question. At least half people use these two words interchangeably as if they were the same thing. So I want us to talk for a minute. Primer. A person or thing that primes, duh. A cap, cylinder, etc., containing a compound that may be exploded, blah, 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 for use of firing a charge of powder. So beginning, this is a beginning thing, right? A first coat or layer of paint, size, etc., given to any surface as a base, sealer, first coat. So think about a primer, it's like the first piece, right, upon which things are added to, correct? Right? It's like the first coat of paint. And so I put a picture here of here's your primer, and bases are added to it, right? Everyone should know that if you've done a PCR, you've designed primers, and the function of the primer is to add the bases to it. It provides a source of the 3' OH upon which subsequent nucleotides are added. Okay? Template from dictionary.com. A gauge or pattern cut out in wood or metal used in woodwork, etc., to help shape something accurately, right? A template, that makes sense. A pattern cut out in a card or plastic and used in crafts to reproduce shapes, right? So all actually relates directly to what it is in DNA. In biochem, the biochem definition, the molecular structure of a compound that serves as a pattern for the production of the molecular structure of another specific compound, right? So your parental strand is your template. So in this, you've got your parental DNA and it's opened up. You have 2 template strands, right? Okay, over and over again, I saw things like telomerase contains its own RNA primer upon which bases are added. Did I ever teach you that? No. I saw DNA polymerase adds an RNA template. I don't know. So really So it could be you're nervous about taking exams. That's okay when I see it a few times. When I see a majority of students doing this, I'm like, either I totally stress you out and you get crazy and start writing crazy things down, or like there's true confusion about function. Telomerase carries its own RNA template, not primer. A lot of people thought it carries a primer. We even had an extra credit talking about how a primer comes in from elsewhere, right? Not with RNA polymerase, with the DNA polymerase action. Remember, you have your the other thing I saw over and over again, this is specific to the lagging strand. No, the leading strand, okay? So here's your leading strand, your parental strand. This is what's extended. There's your RNA polymerase with its own template, and it gets copied as a reverse transcriptase activity, and it gets extended, and then DNA polymerase comes and extends the lagging strand using the newly extended leading strand as its template, okay? So I take this personally because I taught it too. It means I did a bad job if so many people got it wrong. And I even spoke to some people who teach earlier on, and so you get it a little teeny tiny bit in 201. You get it again in 309. I'm your last stop before you're released out into the world. I cannot let you leave with this confusion, especially if you ever want to do anything in science. If you're like, screw this, I'm going into marketing or whatever, then fine, you can confuse it. But if you're going to try to get into grad school, med school, PA school, anything where you might need to demonstrate you understand something as basic as DNA replication, you need to understand these things. So, there you go. That's enough of my rant for today. Please make me proud as you go out into the world. Okay, any questions about that? Does anyone like, no, you always told us this? Yes?
Well, when you say that it's only, like, I got confused, I guess, about the leading versus lagging strand. So
you're lengthening your leading strand so that then DNA polymerase can come in and use that longer template to extend your lagging strand. So yes, the end replication problem comes from removal of that primer in your lagging strand, and that over time again will result in successive shortening, right? But with the activity of telomerase, you're extending your leading strand using the intrinsic RNA template, and then DNA polymerase can come in and copy and extend the lagging strand. Okay, yes?
So would it be technically correct to say that the strand it's adding to is the primer?
Would it be correct to say what?
That the strand that telomerase is adding to is the primer?
That the strand that so you're saying technically this bit here is the primer. Right. Whenever you have a 3' OH that nucleotides are being added to, you can call that a primer because DNA polymerase does not have, now mind you, RNA polymerase doesn't really need a primer, but in the case of DNA polymerase, it can't polymerize from nothing. It can only add. RNA polymerase is different. Yes?
I think I'm a little confused now because I think telomerase has activities on both sides, both strands of DNA, right? Leading and lagging. Because when we have leading strand, even though we just have one primer, when we take out that primer, it's going to extend the lagging strand to.
Yeah. So the way it looks at the end of DNA replication, which is the end replication column, is that right here is where your primer is removed on your lagging strand.
No, we also have one primer in leading strand.
It's in the middle at the origin, remember?
Okay.
Yeah. 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 Okay, let's do project questions after. Do you think it's something that everyone will benefit from, or do you think it's specific to your?
from the image, just CT and house cycle number, cycle correlate.
Okay, so why don't you come meet me for that? Because you don't even need to be looking at CT, you're supposed to be looking at full regulation. But come talk to me after or office hours. Yep. Okay, so does everyone understand this? Were there any more questions about this stuff? Do you see why I'm outraged and having nightmares? Okay. That's good, that means I care, right? I may have nightmares about things that I care about. Maybe? I don't know. Okay, let's move on. Okay, so far what we've talked about. We talked about gene expression, just the big picture of it, and the different types of RNAs that are produced, and how RNA is made in different quantities. You can have tons of RNA being transcribed, or just a little bit, and there are many RNAs on the same gene producing RNA at the same time. Not RNA, RNA polymerase. We talked about similarities and differences of DNA versus RNA, and DNA polymerase versus RNA polymerase, transcription cycle in bacteria, and then we spoke about transcription initiation of Pol2 specifically in eukaryotic cells.
Now that you brought it up, why doesn't RNA polymerase need a 3' hydroxyl to start adding?
It has the ability to synthesize RNA de novo from nothing, whereas DNA polymerase doesn't have that ability. Yeah. So the question was, how come you don't need a 3' OH from a primer with RNA polymerase? It's a different enzyme with different enzymatic functions, so it can synthesize RNA de novo. This is one of its characteristics. Good question. Okay, so this is where we ended. We were talking about how with initiation, you have all these TF2 transcription factors that bind to the promoter and recruit RNA polymerase, and then you have transcription. You also have the function of chromatin remodeling enzymes, because remember, this is eukaryotic genome. You need to move around histones, histone modifying enzymes. And all of this is great, but where is the specificity coming from? It's coming from these transcriptional regulators, activators, or sometimes even repressors that will do other things that bind these regions of DNA far, often very far from the transcription start site. It can be upstream or downstream. It could be in introns, sometimes maybe even in exons. But what happens is mediator helps these transcriptional activators to communicate and aid in assembly with the general transcription factors, resulting in recruitment of RNA polymerase and transcription initiation. That's where we ended on Monday. So a note about supercoiled DNA. So if you think about it, when you're opening up your double helix, you're going to create some torsional stress. You're going to, similar to the kind of things that we dealt with DNA replication, and you end up having this supercoiling, and it's relieved by DNA gyrase in bacteria and topoisomerase in eukaryotes. So just to look at it a little more closely. So just to think about the concept here, for every 10 base pairs of DNA that's opened, the DNA must rotate one turn. Otherwise, it starts to create this supercoil. So here, in this case, it can't rotate because it's fixed. And if you think about it, in a bacterial genome, which is circular, or in a chromosome, you can't rotate your whole chromosome when you open up this teeny tiny 10 base pairs. It's essentially fixed because it's part of this huge, massive chromosome, right? And what you end up having is this supercoiling. Also, you'll get supercoiling from a protein molecule such as RNA polymerase moving along in between the double helix of DNA. So this is where the supercoiling has come from. And it's relieved by DNA gyrase in bacteria and topoisomerase in eukaryotic cells. Okay? Okay, so then you have RNA synthesis, which we're not going to talk about too much. it's a polymerization reaction, similar actually, where you've got this OH group attacking the phosphate group, and then the pyrophosphate goes away, and you get the linkage. Quite similar. The difference is it's happening on the DNA. There's a DNA template strand. It's not this bidirectional thing that you had going on with the DNA polymerase. There's only one. You've got one copy of your template strand, which is going to be similar to your coding strand being created. And again, you've got a magnesium ion in your active site that's involved in catalyzing this reaction and holding everything in the right place. Okay, enough about that. So we're going from gene to protein, right? That's our goal. And there are many steps. And many more steps in eukaryotes than prokaryotes, right? In prokaryotic cells, you've got your DNA, RNA, protein. Not much else going on, right? So transcription, translation, there, then you have your protein. There's a lot more that needs to happen in eukaryotes. You've got introns you have to deal with. You've got capping and polyvenylation that happens. You need to be exported from your nucleus. And then eventually you associate with ribosome and you're translated. Okay, so we're going to talk about the processing for a little bit. Questions? Okay. So transcription is coupled to RNA processing. What does this mean? Kelsey Winters. Kelsey Schwartz. Gonna go through all the Kelseys.
Yeah. Splicing out introns.
Okay, so what do you mean by what does it mean transcription is coupled
Well,
to RNA processing?
To form the functional protein, you need to form the RNA that would translate.
Okay, so to form a functional protein, you need an mRNA that's going to be translated. That's what happens, but what is transcription is coupled to RNA processing mean? Yes?
They happen relatively at the same time.
They happen at the same time. Yeah.
The splicing protein that attached to the polymerase, so it's happening, it will come off, start splicing as it's being transferred.
Right, so splicing proteins actually bind to the polymerase, and so it happens at the same time. As soon as your RNA is exiting your RNA polymerase, it's in close proximity to that C terminal domain, and that's where all these processing factors hang out when they're needed, and it's all based on phosphorylation. Yeah. So that's what we're going to talk about a little bit here. So what do I mean by RNA processing? So prokaryotic RNA, you don't really have processing. If anything, you're having processing of a polyprotein into. So there might be multiple proteins coming out of a transcript, but that's about it. Eukaryotic RNA, you've got this 5' cap that's added. You have splicing, which is going to bring all the coding sequence together, remove introns, and then you have the poly A tail. Okay? It's definitely important to really understand the differences between prokaryotic RNA and eukaryotic RNA. Just in case you need to know it. Like if you're at Burger King ordering lunch and they ask you, quick, which RNA has a poly A tail? Or else you can't have your burger. Okay. Does that ever happen?
Am I even spelling eukaryotic or prokaryotic wrong? I thought it was a K.
They changed it.
They changed
I know, I even still use a K.
Wait, when did they change it?
Oh dear.
Who
When
did
did they
this?
change it?
It's like 2 years
I
ago.
don't know. Actually, and then if you start talking to some people who are very prokaryotic, they get annoyed with the use of prokaryotic versus eukaryotic because it's just so much more than that. So we're going very simple. Let's talk bacteria versus human. Keep it nice and yeah.
So during transcription, it's going from 5' to 3'
prime, right?
Yes.
So the first thing being produced from your RNA is the 3 prime. So the first thing that would happen would be
No, you 5 prime to 3. So
yeah, but RNA production, but that's if you're going 5 prime to 3 prime, then first
let's go back to the picture.
end of the strand would be the 3 prime, right?
The first thing that comes out is your 5 prime. Ah. There. 5 prime to 3 prime change elongation. So the first thing that comes out of RNA polymerase is the 5 prime end. So you're going to say the first processing step would be
Adding 5 prime.
adding 5 prime cap. Right. Exactly. Thank you for introducing that. So 5 prime cap. Okay, so the first thing that's going to happen is addition of the 5 prime cap. And it does happen as soon as the RNA comes out. And so this is, I always love this figure from Albert's. And this really defines what coupling of RNA processing to transcriptional elongation means, right? So you've got your RNA polymerase doing transcription and your 5 prime end of RNAs popping out the end here, right? And here's your C-terminal domain of RNA polymerase. And I keep harping on the importance of this domain. Really make sure you understand it. I don't want people telling you this is RNA. It's not. It's part of your RNA polymerase enzyme, okay? And what happens is all of these processing factors bind to the C-terminal domain, and it's dependent upon certain types of phosphorylation. So notice there are different phosphate groups being added at different places. Remember, phosphorylation changes the shape of things and is going to control recruitment of different proteins and protein-protein interactions. So first thing that happens is you get phosphorylation on serine 5, and that results in recruitment of cabin factors just in time for the 5' end to be coming out and being right next to that C-terminal domain. And then you'll have, and so what this is showing is the cap binding protein, actually. And then you'll have splicing proteins recruited with a different kind of phosphorylation. So here you see 2 and 5 phosphorylation, and then you're going to have, and that, see, it's come out that much more, and so you're starting to have access to introns, right, and splice sites. And then you'll have a different kind of phosphorylation, a different combination of phosphorylation that's going to result in recruitment of 3' end processing proteins. And that's going to be as you get towards the 3' end of your RNA. So this is what we mean when we're talking about coupling of mRNA processing to elongation. It's actually happening at the same time because all of these proteins are associated with RNA polymerase. Okay? Any thoughts? Okay, so step 1, addition of 5' cap. This is a way, so you always have to ask yourself, why? Why 5' cap? I mean, you can memorize the list if you want, but if you want to really enjoy this class, think about why, right? So why would you want to add a 5' cap? I kind of say it up there. A way for the cell to distinguish mRNA from other RNAs, right? What other RNAs are there? Yeah. Okay, viral RNAs. What other RNAs other than some viral RNAs are very similar, actually, to our mRNAs. Yes. Ribosomal RNAs. Yep. tRNAs, transfer RNAs, small nuclear RNAs. So yeah, RNAs that are not coded, RNAs that are not mRNAs, RNAs that do not need to what? Be translated. So do they necessarily need to be treated the same way as an RNA that's going to be translated? No, because a lot of what needs to happen here is you need to get your RNA ready to be translated. So that means that you need to be able to export it from the nucleus. You need it to be able to associate with the ribosome. These are all very specific things that only need to happen to mRNA. Your tRNA needs to associate with ribosome, but in a very different way. Your ribosomal RNAs need to associate with the ribosome, but in a very different way, and actually in a very different place. So it's different. So the 5' cap is one way for the cell to distinguish mRNA from other RNAs, and it helps with processing, export, and translation. Okay? Yes?
So since it helps the mRNA export to the nucleus, does tRNA have a different mechanism in which it's able to export?
Yeah, so one doesn't exclude the other. So this is the mechanism for export of mRNAs out, and a lot of it is because you need a way to quality control what mRNAs get out of the nucleus. So one way is to have all these different proteins that bind to certain elements that come from this processing, and that kind of shows that this RNA is mature and ready to associate with the ribosome, and also therapeutically bind to the ribosome. Export of tRNAs out of the nucleus. How does that happen? Gotta get out somehow. I bet you there are specific proteins that help to get them out, but I have no idea what they are. I'm sure it's known. Gotta be known, just not by me. Yep. Good question. So 5' capping occurs after about 25 nucleotides are synthesized. So fairly early on in transcription elongation. And so what do I mean by 5' cap? Is it like a little hat with 5 on it? Not really. Right, so here's your RNA, the blue, and your 5' cap is 7-methylguanosine. And there's a specific reaction involved with capping. So there's 3 main enzymes. There's a phosphatase. What's a phosphatase going to do? Cut off a phosphate group. Ideally not all of them, because we're talking about RNA. If you cut off all your phosphate groups, you don't have RNA anymore, right? Since that's how bases are connected. Right, so what it does is it cuts off, so you've got this is your first base here on your 5' end, and there's 3 phosphate groups. It removes 1, your final phosphate groups. There's a guanyl transferase. What do you think a guanyl transferase might do? I know a lot of people know it because I hear lots of murmuring. Transfer what? Guanine. Transfer guanine. So transfer a GTP, actually. And then it adds it on here. So you got, it's a similar kind of reaction. If we go back, you can kind of see, well, no, you can't. It's a similar kind of reaction as you have with DNA polymerase, where you have an OH attacking, I believe. Maybe not. I might be wrong. Then you have a methyl transferase. What do you think that does? Adds a methyl group. Great, you guys have learned this before. Wait a minute, why is there a methyl transferase involved with this? Isn't that only involved with histone methylation or DNA methylation? What? Right, there's a lot of methyl transferases that work on a lot of different things. So those, right? So you've got, so there's your phosphatase working. There's your guanyl transferase working right there, and then there's your methyl transferase working, and you get methylation there. And that's how you get your 7-methylguanosine 5' cap. Okay, very exciting, right? I would never ask you to repeat these steps back to me because it's not useful, but the reason I put them up is because I think you should know what all those enzymes do automatically. Like at this point in your career, you shouldn't have to think about what a phosphatase is, and you shouldn't have to think about a methyl transferase. You might need to think about guanyl, what guanyl is, but transferase should at least, you'd be like transfer something, right? So all 3 of these enzymes bind RNA polymerase to C-terminal domain, when it's phosphorylated as serine 5. Right? So that's right there. There's serine 5, there's your C-terminal domain, and capping factor showing one thing for simplicity, but they bind to this shape. Okay? Oops. Okay, so what transcription factor did we learn about that's responsible for this phosphorylation of the C-terminal domain that's required for recruitment of these enzymes? Yes? Okay, so a kinase, maybe, something with a kinase domain, perhaps. What transcription factor did we learn about just the other day that I said is required for phosphorylation of the C-terminal domain, which is involved in recruitment of processing factors? Remember, yes? TF2H. Remember, I said TF2H, it binds, it has helicase activity, it also has a kinase domain, likely, and is able to phosphorylate the C-terminal domain. Trying to piece this all together here, right? Absolutely. Okay. So, I kind of already discussed this. Why aren't other RNAs capped? Well, I did kind of discuss it, and I didn't actually. Take that all back. I discussed it in my head while I was just talking to you. There's something wrong with me. Yes?
You said that they don't need to be, but we don't.
Okay, so why aren't other RNAs capped? Yes?
Never mind. I was gonna
Do
say
you want to
that.
discuss with your group?
Group?
Anyone?
Someone, come on. Yeah. Yeah, I guess no one wants to discuss.
Okay. Yes?
Would it be because they're not being translated, so they don't need
Well, so you're giving me a result and a reasoning why. But why, like, mechanistically, why aren't other RNAs, not mRNAs, like tRNAs, let's say, why aren't they capped? Yes?
Maybe they don't need to be translated. That's why.
So you're giving me a reason why mRNAs are, right, so they're not translated, right? Yes?
We don't have the association with Paul II tail.
Right. They don't have association with Paul II tail. Why don't they have an association with the Paul II tail? Yes?
They use a different polymerase.
Right. They use a different RNA polymerase. And those only RNA Paul II has the C-terminal domain. So now, would that mean that these are only processed because they add whatever? Yeah. So only RNA Pol II has a C-terminal domain. So only RNA Pol II has the ability to bind in this regulated fashion to all these processing factors and process mRNAs. Yes.
Would the other RNAs have your own post-translational modifications?
Do the other RNAs have modifications? Basically, is your question. I'm sure they must have something, but I'm not sure. I have to think about it because we will touch a little bit more on translation than I have in the past, because I always feel like that's missing. And I know there are a fair number of things that need to happen to tRNAs to become tRNAs, right? Because they don't start out at a tRNA that's ready to associate with a ribosome and add an amino acid, right? But I don't know off the top of my head what they're doing. And I think it's long after they're transcribed when they do get modified. Good question. Any other questions? Isn't that cool though? Who finds that to be very cool? Who's a geek? Yay for geeks. Okay, who's like, this is so uncool? I wish I was at the mall. I wish I was at the mall. Whenever it gets nice outside, I want to go dress shopping. Right, who wants to go dress shopping? Should we just leave and go dress? Should we just leave and go dress shopping? We've got like 15 minutes until the end of class. We just get out early, run up to the mall, buy some dresses. Guys, come wear dresses. I have a friend who wears dresses. He looks better than me. Anyway.
Splicing mechanism only binds RNA Pol II as well. Or can that bind to other
The splicing factors, do they bind to other RNA polymerases? That goes back to the same thing. It binds to the RNA C-terminal domain. And so any RNA polymerase that doesn't have a C-terminal domain can't bind to the splicing factors, and they're likely to not be spliced. Right. Good association. Could say the same thing for the polydenylation and those factors. Okay, very good. Very good. How are we doing? Okay. It's supposed to be like 91 degrees today. Yeah. But this isn't it. We're going to get nice and cool again next week. Okay. Splicing. So this is where we're going to end. We're going to talk for a minute about splicing, and that'll be that. My friend who's in undergrad right now, some people take their time to get there, right? And he's rocking it. He was like, I think classes are always so much easier after spring break because I think the faculty is just done. And I'm like, totally. You're totally right. I think we're all done now. Okay. So this is just looking at two types of genes and how things can be organized. You can have a really small gene with the exons are in red and the introns are in orange, where with small introns and small exons. And then you can have these large genes, right, with many, many, many small exons. Exons generally tend to be very, very small, and introns tend to be much larger. I'd say that's a rule that you can stick with. And the removal of the introns will result in a much smaller mRNA than the gene. I think we can all agree on that. Not much, much smaller in this situation, but there's a diversity in what genes look like. Yeah?
Is there a general consensus now about what introns actually do in their functions?
I have to sneeze. It's not going to come out. What do introns do? Are we going to talk about evolution? Because I guess we kind of have to in this discussion, right?
I mean, there was a lot of debate about what their actual role was.
Right, so I think there's a lot of things that happen within introns. You can have transcription factor binding sites in introns. You can have sometimes microRNAs that are generated from intronic whatever RNA sequences. But then if we want to talk about evolution and how we've survived as a species, it makes sense that if you kind of have these small bits of important information peppered throughout this big, long mess of RNA that's going to be spliced out, and you've got all these, you've got constant insults to your genome, and let's not forget things like retro elements, which I'm really sad I probably will not even get to in this class. There should be a whole class on retro elements. But they like to jump around in our genomes and mess with things, which sometimes it's good because it results in expansion of different genes, but sometimes not so good if it disrupts genes or regulatory regions. I had a long conversation with Dr. Masters about this, where we were talking about genome size and how sometimes a really large genome correlates with higher activity of these retro elements because they can jump around just fine and not kill off a species by damaging their DNA. My guess is this is kind of to keep things kind of stabilize the genetic information that we need to make coding regions or coding proteins. I don't know. What are your thoughts?
That's not everything I've heard of.
Okay, yeah. So it's a lot of blah blah, hand waving, blah blah blah. I'm sure there's more people who really study this.
So when someone says that a species genome is X% of another species genome, is that the entire genome or is that just coding regions?
Oh, when somebody says a species genome is has X identity to another species genome, is that whole genome or coding region? I would think whole genome, but I have no idea what they base that on, because only the most important parts of a gene are likely to be conserved. I don't know. microphone now. Yes.
In that picture that you had with the bacteria eukaryotic mRNA, so when the non-coded sequences are cut up, does it in the bacteria does it make 3 separate proteins or is it?
Yeah, so this is like a polycistronic mRNA, which is going to have 3 different proteins. So it's not actually splicing out of different parts of genes to make one protein. It's going to code for 3. This is a common way that bacteria regulate genes that are related. Is they'll have one, like when you've learned about the lac operon and stuff like that. Sometimes you'll have one regulatory region in the DNA, which will start transcription of multiple genes in one long RNA, which will then become multiple proteins involved in one function.
It's sort of just semantics. Is that one gene or is that 3 genes?
These are 3 genes.
Okay.
Yeah. The question is that 1 gene or 3 genes? It's actually an operon.
Okay.
Right. So who took Med Micro? Did you learn about operons in Med Micro? And you probably learned about it in 309. We're going to learn about it again a little bit. But operons are just ways of organizing groups of related genes under the control of one promoter. So it's multiple genes. You want to know really what defines a gene.
Yeah.
I'd say yeah.
Because they used to say like 1 gene, 1 protein, but then there's all sorts of exceptions to that.
Yeah.
Now it's like,
I think it's 1 gene if it. Got it. Let's not go there. Okay. Yes.
So then in bacteria are they non-coded region cut up or do not?
It's not that they're cut out. It's that you're going to have it's not like splicing. It's not that they're It's not a splicing reaction that cuts them out. You're going to have cleavage. I think you might have translation straight through and then cleavage of proteins. Am I right? I don't remember.
no, there's probably a sound. What would that be?
Depends. Give me a minute to think because I once used a plasmid that expressed multiple proteins from one promoter, and I believe they did all have start and stops, but they have internal ribosome entry sites. Different story. I'm not going to take you too far off topic. Don't even think of this as similar. Actually, probably them putting this in it makes it more complicated. This is from Alberts. But just think there's the individual start and stops for each protein, so that makes them individual proteins. I'll get back to you on exactly how that happens if you want to know. We'll probably do a little comparing of that when we talk about translation. Good. Other questions? Questions? Everyone's awake. Great. Okay, splicing. Okay, here's your splicing reaction. Okay, so you have your exon, and you have your exon, and here's your intron. And basically, within your intron sequence, there's this adenine that attacks the 5' splice site, which basically cuts the RNA right there and joins it together, and leaves this free 3' OH, which then reacts with the start of the next exon and joins them together, and you get release of this lariat structure, total lariat, and joining of your 2 exons, and that's the reaction. That's the most basic form of it. Yes.
What enzyme is responsible for joining the 2 pieces?
I think it's chemistry. So what happens is, I think it's chemistry. What happens, and we're going to go much more deeply, this is just an introduction to splicing. There are all these splicing factors that help to bring things close together. So when you have something like a free 3' OH, it can attack the whatever's over here.
So this would happen at some low rate without the presence of an enzyme.
So the question is, would this happen at a low rate without the presence of an enzyme? So do things automatically splice? I think it depends on how close together things are, because you need to notice how this is leaked out. You actually need to bring things close enough together so electrons can move. Huh?
Some relate that would happen without enzyme.
I don't know. Would it?
I think I read that.
So maybe at some low rate this might happen without an enzyme from the front row. I guess it really does depend on the structure of the gene. I can guess with in ideal conditions where you have a not so huge intron, it seems logical. Yes.
And the other mechanisms for which the intron splice, or does it always form this lariat intron?
Does it always form this lariat, or are there other mechanisms? Well, let's go through the whole mechanism, which we'll do on Friday, and we'll see if there's anything else, but this seems to be what sticks in mind. Yes?
How can this happen between two or more introns for alternative splicing?
Huh?
How can this happen between two or more introns for alternative splicing?
How does this happen with alternative splicing? How does this fit in, right? It's about splice site recognition. So sometimes you'll have splice sites, you can think of as having different, so this is a splice site, right? So sometimes you'll have a very strong splice site that will be recognized readily, and sometimes there'll be a weaker one, and under certain conditions, the weaker one will be able to function. We'll get into it. Yep. Oh, that was forward. So if you remove your introns, would you remove your background cap and foliate tail? Very nice way to think about it, but this is actually only showing part of the gene. It's actually outside that region where the capping and the foliate tail are. Yep. Okay, are we good for today? Everyone go out and dance in the sunshine? Or come to rest of the Yeah. I believe my presentation.